Okay, so this video is regarding the remaining contents of the transport in plants. Okay, so this will be going. It will be recorded, right? So you can view it any point of time. So morning, good afternoon, Arsha. So morning session we discussed about I introduced the topic of this water potential, right? So what exactly the water potential, and and this is very interesting because the movement of water from within the shorter distances would be controlled by this phenomena, right? These are the concepts behind them. So how these how these what do you say, all these things are related. For example, water potential, solute potential, pressure potential, potential due to gravity, right? So how what are the positive forces and what are the negative forces also we will be talking in it. Okay, so let's start begin with. Okay. So within this water potential, right, we already talked about this water potential. So this water potential equal to pressure potential, solute potential plus pressure potential, right? So here, how can we draw relationship? How do we understand this? How can we understand this? Water potential, right, will be high if its solute potential is less. It means if you take the water, a sample of water, and if it contains more number of solutes, right? If it contains more number of solutes, it means water molecules are less. In the sense, the kinetic energy of the water would be less. It means the water potential is less. So that's why we have pure water. For pure water, NTP, at normal temperature and pressure, NTP, you will see that pure water has more or high water potential compared to other samples. So the solutes which decides the water potential. So whenever there is a low water potential, water always tend to move from higher water potential. I'll write WP, higher water potential to the lower water potential. Okay, so it means, for example, if a cell or any substance, right, for that matter, any substance which has less water potential has more absorption phenomena. So it will this if you consider this as an object, some substance or a cell, so it has low water potential. So it tends to absorb lot of water in it. So more absorption would be taking place. So in this way also you can understand this. More absorption by the cell when its water potential is less. Why? Because always water tends to move from higher water potential to the lower water potential. And now we know why water potential is less in a cell, because it has more solutes. And outside environment you have less number of solutes particles. So in that way we can figure it out the water potential relationship, right? Yeah, good afternoon. Clear, right? So that is one of the aspect. So these are very confusing actually. If these things comes in a examination, what you can do is write this psi w, psi p, and psi s, and you try to recollect that. No, this water potential lowering or higher is depending upon this this solute potential. If more number of solutes are there, obviously the water potential comes down. So in that way, these statements can be asked as correct or incorrect statements also. So you should be very much careful. How much ever we try 100, we revise 100 times, it will not be useful. But you can write like this, right? We have write for linkage concept, right? In genetics, like that you can write some equations, right? Directly proportional or indirectly proportional. Like that, if you write, then it will be very easy to recollect. Okay. So next aspect is solute potential, right? Solute potential I already discussed. So solute potential is high in the sense it will have less water potential and it will have more absorption. It can absorb lot of water. Why? Because it has more solute potential here, right? So if you consider in this, it has more. So how can we talk about how can we reframe or define this osmotic potential or solute potential in the chemical potential of the pure water due to the presence of solid particles in it. So due to presence of solid particles in it, right, the chemical potential of water would be decreased. So that is nothing but solute potential. So once the solute potential is, it has to be always comes in a negative values. This solute potential. Okay. So it always have a negative value when we calculate, right? So next one is pressure potential or turgor potential or hydrostatic pressure or pressure potential. So the next one what we are talking about is pressure potential. So this water potential and solute potential is understood. Water potential and solute potential. So we completed water potential, solute potential. Now we'll be talking about the pressure potential. So okay, fine, good. So next we'll talk about the pressure potential. So how can we understand this pressure potential? So pressure potential we can understand by any living cell you take. Okay, living cell is there, right? So when it is placed in an hypotonic solution, okay, what is it? Hypotonic solution. Hypotonic means concentration is less. Okay, waters tend to enter into the cell by osmosis. So when we place this cell into, if you consider this is a cell, okay, consider this is hypotonic solution. So water tends to move inside the cell. Okay, so pressure develops inside the cell, right? So because the water is entering, so this pressure presses the protoplasm against the cell wall, right? So now if you measure the cell, the cell length or the cell size. So before you place it in the hypotonic solution, the cell size would be like this. Okay. After placing into the hypotonic solution, the water enters into it, and finally it becomes turgid like this. Turgid. So this is osmosis phenomena where the water tends to enter into this cell because cell has more solutes and water has less solutes, because this is hypotonic solution, means number of solute particles would be less. You would have studied in chemistry also, hypotonic, isotonic, hypotonic solution. So when we place it in hypotonic solution, what happens? Water tends to move into the cell. Once the, for example, this is a room, right? Suddenly tsunami came or some water is trying to pump inside. So what happens? Assume that this is made up of elastic walls. Then when the water is coming inside, it would push the walls and the wall shape would be changed. And the plant shape is actually maintained by this process, turgid process. Understood? So this is pressure potential. Pressure potential is something positive. Okay? Is that clear? Or we can call this pressure potential as also called as the pressure, what we can call it as turgor pressure. Okay. So this is the relationship between different what do you say, water potential and the other solute potential and pressure potential. Okay. So pressure potential will be usually a positive value would come, where you will be finding solute potential would be always negative values would come when we try to calculate the water potential of a given solution. Okay, so that is about the relationship, right? So let's move to the next one. So even the pressure potential, so by applying certain pressure, right, even it increases the water potential. So that is one of the important aspect. So why this pressure potential is positive is one of the aspect is that, by applying certain pressure, even it increases the water potential. So it is actually adding to the water potential. That's why this pressure potential is something a positive value. But solute potential is always negative. Why? By the presence of solute, it eventually decreases the water potential. So that's why it is negative. So it is inversely proportional and this is directly proportional. So that in that way you can draw a final conclusion. So water potential, if you take, it's directly proportional to pressure potential and inversely proportional to solute potential. If more water potential means less solute particles. So this thing you need to remember. So this is the single agenda where I conducted this second class actually. Okay. So these things would give more clarity. So when we talk about osmosis also can be good afternoon, Akshay. So osmosis also can be actually can be studied in two ways. One is osmosis. If you see, it is a simple diffusion of water across differentially or semi-permeable membrane. It means it allows only certain molecules. Okay. So here, this if you see the worksheet that I provided, so the osmosis can be actually understood by two things. One is pressure gradient and concentration gradient. So gradient is something from higher to lower or lower to higher. In the same way, pressure also, more pressure to lower pressure. So always the air try to move from higher pressure area to the lower pressure area, and then concentration, higher concentration to lower concentration. If you see this also, higher concentration to lower concentration. So likewise, you can see this osmosis where water potential, especially the pressure gradient. So when the pressure is applied, the pressure gradient is created within a system during this osmosis. It eventually increases the water potential. That's why this water potential or this pressure potential would be positive, and it eventually it eventually increases the water potential capacity of the water. So you see this osmosis, So osmosis is very simple thing. That's not an issue. Okay. So osmosis, where we have a simple example also, a simple diagram also given in NCERT, right? You need to understand this osmosis by two factors. That is, one is pressure gradient, right, and then concentration gradient. So the net direction and rate of osmosis. For example, if you take chamber A and chamber B, right, the movement of molecules from A to B or B to AR, the net direction would be actually dependent upon the pressure gradient as well as concentration gradient. Okay, so water will move from its higher chemical potential concentration to its lower chemical concentration. I told now, higher water potential to the lower water potential, always water tends to move in that direction. Higher water potential, in the sense you will have less solutes. So this is lower water potential and this is higher water potential or chemical potential. Chemical potential, right? So the direction, net movement will be in this way, A to B. Are you getting? So in this way, concentration also you can see here. If you see here, this is how the water, chemical potential will be talking about, right? We can take this as an example, right? So until it attains certain equilibrium, this transfer would be taking place. Okay. So when it will attain equilibrium, when the water potentials of both would be in equilibrium, it means both water potentials should be equal. Then only we can get this. Okay. So you would have discussed about osmosis by using some sugar solution, sucrose solution kept in a funnel, and we will be demarcating its level, and we will be keeping in the water, and the water would diffuse. So, and what we will do is we will try to apply certain pressure to one of the funnel. It is a small experiment. It's a self-explanatory also. So based upon the pressure that we apply, even the level of water that is seen in one of the chamber would be increasing. It means the simple way to remember is, when the pressure is applied to a water, its water potential also increases. So that water tends to move. So that is one of the aspects, that is osmotic pressure was explained in one of the diagrams, that funnel experiment, right? When we apply pressure, how this pressure, osmotic pressure, would actually increase the level of absorption, the water absorption, okay, within the funnels. So that you need to once go through. It's a self-explanatory, that's not a problem. Yeah. So let's move to the other important aspects. Okay, quickly we'll go. And then when we talk about plasmolysis, if you see this plasmolysis, I gave three different things in plasmolysis. I gave it in flowchart in your material, right? So if you see that plasmolysis, okay, if you see the plasmolysis, so plasmolysis can be of can be understood by three important processes actually within the plasmolysis, right? Three important phenomena will be there, right? What exactly is the plasmolysis? It's an actually behavior of the plant cells, right, with regard to the movement of water, right? So there are plant cells will be there, and the plant cell would behave, or it certainly, either it would what do you say, it will expel the water or it will absorb. So this plant cell behavior or tissue behavior based upon the availability solution, the surrounding solution. So if you have a plant cell like this. So this plant cell would behave, the same plant cell would behave in a three different manner based upon its external solution. So if you take this as external solution, okay, so under three different conditions, the cell would behave in a three different ways. So that is what I gave in worksheet also. What are those? We'll try to discuss right now. So one is placing the cell in isotonic solution. So there are three different behaviors it shows. That is, it means it becomes flaccid. Cell becomes flaccid. Okay, flaccid, and cell becomes turgid. Okay, turgid, and cell becomes plasmolyzed. These are the three different phenomena, three different behaviors that cell or tissues will show. When it will show, let's see. So we are talking about the surrounding solution. Huh? So in which type of solution this phenomena would be seen? Let us see. So first one is, let's consider A. In A type of condition, what you will find is, this is isotonic solution. Isotonic solution. Isotonic means, iso means the concentration of the water molecules present inside the system and the outside the system remains almost the same. So they will be in equilibrium. Okay, when they are in equilibrium, what happens is there won't be any much transfer of any transfer of water molecules will not be seen, right? Are you getting? Understood this point? This is very simple actually, this concept, but let's try to understand. So in a isotonic solution where the concentration is, for example, the external solution balances with the osmotic pressure of the cytoplasm. Okay, so osmotic pressure which is present inside the cell and the external solution would be, osmotic pressure would be the same. So that makes the isotonic solution. But what happens to the hypotonic solution? In hypotonic solution, you'll see external solution is more dilute compared to the cytoplasm. So what we figured out here is, we will talk in terms of cytoplasm. So in cytoplasm, you will find lot of solutes, right? So cell organelles, we have a lot of molecules, we have water. Less water may contain, and you will have a lot of different organic molecules. That makes it more number of solutes. But here you will have less number of solutes. So when the less number of solutes means, now you have to quickly recollect. Here we have less number of solute particles. So it will have higher water potential. When higher water potential is there, it tends to move from higher water potential to the lower water potential. But here, when it will be taking place during hypotonic solution. So in hypotonic solution, this would takes place, right? Lower solute particles and higher the water potential, so that the water molecules tend to move inside the cell and makes it turgid, hypotonic. And this is isotonic. Isotonic means the concentration and pressure gradients of the cell and the external system solution would be in equilibrium. So there won't be any transfer of water. So it remains just like that. So that's what it attains the flaccid kind of structure. In stomatal opening, the pore opening, stomatal opening and closing, this concept would come, flaccid and turgidity. Right? So this is hypotonic solution where the less concentration, and then you have a lot of solute particles within the cytoplasm. So the water tends to move inside and the cell would bulge. We have seen the turgidity, pressure, right? Pressure, same concept. Because it increases, it pushes the cell wall and it pushes the cell over in all direction and it becomes turgid. And the last one is hypertonic solution. Okay, hypertonic solution. In hypertonic solution, what happens? Right, the number of solute particles present inside the cell would be less, cytoplasm would be the less, and the outside would be more. So more the solute particles, less the water potential. Are you getting? So when the less the water potential means, and more the solute particles means, where exactly more solute particles? Outside the cell. Solution we have more solute particles. And inside the cell, comparatively, you have less solute particles compared to outside. So you have more water molecules inside. So it has more water potential. So the water tends to move from inside the cell to outside its surroundings. When water is lost by the cell cytoplasm, what happens is it becomes plasmolyzed. You can take a grape and you can put it in different salt solution or sugar solution and a normal water. Then you can observe how that grape would actually behave. Is it clear? These three things. Now, is it clear? I explained three phenomena, right? So if you have certain notes, na, you can actually keep in this way, right? Isotonic, flaccid; hypotonic, turgid; plasmolyzed cells where hypertonic. Okay, the cell remains in the same shape and size, and here, okay, let's put this. And here it bulges, becomes turgid, and here it loses its water and becomes plasmolyzed. And here you try to write the water potential relation and solute potential relation and pressure potential relation also. So here water, how can we write here is very clearly. So you can draw a line here, and you can write here inside the cytoplasm and outside. Outside in the sense solution. Okay. So inside the cytoplasm, during flaccid, both are in equilibrium. Isona, so equal. And in turgidity, hypertonic solution means what's happening here? Here what's happening is nothing but equal in the sense like water potential of the cytoplasm and the water potential of outside is equal like this. Okay, and the next one you can do is in the turgidity cell, or you have hypertonic solution. Hypertonic solution means low concentration. Low concentration means high water potential, low solute particles. So inside the cell you will have more solute particles, right? So you can write more solute potential, less water potential. And outside you have less solute potential, more water potential. So water tends to move from higher water potential to the lower water potential. Clear? And then last one, plasmolyzed cell. How can you write that? Reverse. Inside the cell you have less solute particles but high water potential, and outside the cell, it means in solution you have more number of solute particles, so high solute potential, and you have less water potential, and the water tends to move from higher water potential to the lower water potential and makes the cell plasmolyzed. So the cell contents would be coming outside. So this is the movement of water. Okay. So this is equilibrium. This is the concept. So based upon this, you may get different questions. So they may give diagram and they may ask, no, water potential is high, solute potential is less. Whether this cell will be in plasmolyzed cell or it is a turgid cell or flaccid cell, right? So that is the concept. So let's finish this. Is it clear, everyone? Whoever listening? In NCERT, they have given this, so you can go through once. And the last type of osmosis, right? Diffusion is nothing but imbibition. Imbibition, I already shown a demo also. What is imbibition? Is a special type of diffusion, right? I have shown these seeds, right? How they imbibe the water content in it, because seed will contain lot of solutes. Now you can apply this thing to anything. But remember, if this is a plastic, this absorption phenomena, imbibition by solid particles, even depends upon the nature of the imbibant, nature of the substance which is trying to absorb the water. Plastic cannot absorb water, but a seed can absorb water, wood can absorb water. So even the absorption or rate of absorption or rate of imbibition is even dependent upon nature of the substance that is absorbing. Is that clear? That's it. So with this we completed short distance water transport. Okay. So now let's enter into the, once the short distance water transport is done, then we can really understand the long distance transport, because these are all small distance traveling, and then that only which gives more impact to the transport of the water. So we will be talking here different theories, different aspects we will be covering in this. So let's see what are the different things we can cover in this long distance transport of water. Okay. So long distance of water or translocation, we will say it can be of bulk movement. If you see this osmosis, right, you will be having, through the semi-permeable membrane, it selectively sends certain type of molecules. If you take diffusion again, the rate of diffusion is depending upon different factors, right? These are all the important things for the NEET. What are the things that are factors affecting the rate of diffusion, rate of osmosis? All these things are important, right? Size of the particle, concentration of the particle, type of the particle, right? Nature of the particle. All these things would affect the diffusion. But when it comes to bulk flow, what happens is it is as good as this. If you go to your home, you will be allowing only people who you knew. But when it comes to school now, 1 or 2 gates, right? All the people would enter into the school. Okay, so it means bulk flow. All once first standard to 12th standard, every student would enter to the same gates and exit to the same gates. But when it comes to our homes, only few people who we knew selectively we will allow them. Selective permeability. So that's what's the difference between the bulk flow and the remaining, the short distance transport. So this is a bulk flow is nothing but it will allow translocate this water, minerals and food generally moves as a mass, right? Or a mass system or a bulk flow system, right? So this mass flow is movement of substance in a bulk or masses from one point to the another, so that as a result pressure differences would be created from one point to other point. Is that clear? So the pressure differences would create because and then this will keep on moving. So from higher pressure to lower pressure, this movement would be taken place. Okay. So unlike this diffusion, they can independently move with respect to even without concern about this concentration, size of the particles, etc. So that is the important aspect of this bulk flow when compared to diffusion. Okay. And even the bulk flow can be achieved through a positive hydrostatic pressure gradient, right? Or a negative hydrostatic pressure gradient. So either we can create this bulk flow movement either by pushing, right? Pushing, that is nothing but the positive kind of a force, or pulling force, hydrostatic, negatively hydrostatic pressure. It means suction, like a vacuum suction pipe, like we can, or the pump, handset pump will be there now, where it would be sucking the water from the ground. So either negatively with this hydrostatic force or the positively. So one is either pushing force or pulling force, right? So, but ultimately what it does is it translocates the food particles through this xylem and phloem tissue system, right? So if you would have remembered this, morphology, anatomy, when we talked about anatomy, we talked about this xylem, phloem arrangement in leaf, root, and stem, what do you say, and how they arranged and etc. Right? If you would have remembered the parts, right? If you take the roots, right? So the water tends to enter into the vascular vessels, right? From root hair to the epidermis to the cortex to the endodermis, and then it enters into the xylem vessels, right? This is a flow chart, right? So I'm rubbing this. I hope you note it down, or you can come back and you can visit this. Okay. So this is very important relationship. Were not given in this format. If you remember in this format, it would be useful. Okay. So what we are discussing? This right, from the root hairs. So you would have been remembering. So root hairs present on the epidermis, right? So root hairs, epidermis, and then you will be finding cortex, and then endodermal cells will be there. Okay, and then into xylem vessels. So this is a mode of transmission, right? So here now the question is, how do plants absorb water? So there would be two different pathways that are present in the system, right? Plant system. So before that, you need to really remember the anatomy of the root, right? If you remember the anatomy of the root, you will be having root hair, you will be having epidermis, cortex, endodermis, which contains Casparian stripes, and then you will be having pericycle. Within the pericycle, which is closely arranged xylem tissue. So it is given in NCERT, as well as I have given in what is this PPT, right? So please access that file PPT so that it will be more useful. So if you see that the green bars and pink bars that was given in PPT, it shows the movement of water molecules. So this water plant absorbs water in two mechanisms. One is hypoplastic pathway and symplastic pathway. The next one is symplastic pathway. What exactly this pathways talks about? Okay. So if you see this hypoplastic or symplastic pathways, these are two different pathways where the water actually enters into the xylem vessels, right? So if you see this, hypoplastic pathway, So in hypoplastic system of pathway, what happens is, even in NCERT also it is given clearly, right? So you will be having epidermis, right? So you have root hairs and epidermis. Epidermal cells will be there, and you will be having cortex, right? Cortex will be there, and then you will be finding endodermis, and then at the pericycle you will be finding xylem vessels. So water has to enter right from root hairs to to the xylem. So how will it will in two modes it will go. What is the first mode? First mode is we are talking about apoplastic movement. Okay, apoplastic movement. Where in apoplastic movement, the adjacent cell walls, that would be continuous throughout the plant. So if you take this, this cell wall, the water would actually move within this cell wall components, right? Continuously it will move. Only it will not move at the Casparian stripes. So you will find Casparian stripes at the endodermis. So this is cortex, right? This is cortex, and you have endodermis, right? So to the endodermis, you will be finding this Casparian stripes. So except here, right? So the water tends to move. Let's assume this black is water. So it sorry, this is not black. So this water would move like this, move like this, move like this. But wherever at the Casparian thickenings, right? Where exactly the Casparian stripes are there, it will not move towards the walls of the endodermis. Then it will enter into the cytoplasm, and then it will then enter into the xylem. So this movement of the water molecules through the cell, exclusively through the intracellular spaces and walls of the cells is called apoplastic movement of the water. In your NCERT, what they gave is the colors. They mentioned in a reverse format. I think your botany teacher might have been informed you last year. There was little correction in the diagram. You please see to it. Okay. So now we'll be talking about the diagram of 11.6, figure 11.6. Okay. So go through it once. Okay. Fine. And the next one is symplastic movement. If you consider this is the black is water, water would be moving through the interconnected protoplasm. So here you will be finding interconnected protoplasm. So these two protoplasms are interconnected to the plasmodesmata. So this plasmodesmata would act as a bridge for two cells. So within this, this is plasmodesmata. So within this plasmodesmata, this water tends to move like this. Right? So these are the two important mechanisms that water would enter into the cells. So those diagrams already given. Is more than this, it's not that much important. That's it. So these are the two aspects you need to cover: symplastic and apoplastic pathway. Okay, especially where exactly this symplastic and apoplastic are taking turns, in the sense like they are not traveling through the cell wall. Where exactly at the endodermis, where Casparian thickenings or Casparian stripes are present, that you need to focus. I gave very clearly in the PPT as well. And the next one is in the long distance water transport or bulk flow system. This water movement can be of through the root pressure or transpiration pull. I told now one is pushing force and other is pulling force, negative force, and one is positive force. That is root pressure, and the negative hydrostatic force, that is transpiration pull. So these are the two aspects we need to understand. Okay. So water movement is upward is because of the root pressure. Root pressure in the sense like what exactly the root pressure? In a vascular tissue of the roots, the water flows, right? Flows and increases its pressure inside the xylem. So the xylem is there, right? So in the root system, this water tends to move inside the xylem and it increases the pressure, and it creates certain positive pressure in the root, and that is called root pressure, and can be responsible for the pushing of water to the smaller heights, not too much. Okay, smaller heights. Okay, upward movement of water. When we talk, we have both root pressure and we have transpirational pull. Transpirational pull. So this is something negative hydrostatic force. This is positive hydrostatic force. Hydrostatic pressure. So when the water is trying to enter into the xylem, right, it creates pressure and then it tries to push, right, push the contents of the xylem and other minerals at certain levels, certain height, right. So this positive pressure is always certain, it adds up to the movement of the water, isn't it? And how can you really how can you conclude this? You can conclude this by a simple experiment. You can cut the stem, right? You can cut the stem and you can put it in some colored water, so that you can see that the water would be coming out of the stem cuttings. Why? Because this water is trying to move from the root system to the different parts of the body, and this process is called guttation. It means this is one of the type of loss of water, right, from the excess water within the plant system also can be moved out of the plant body by guttation, right, and and through evaporation also. So what affects this root pressure? So if you see this root pressure, right, excess water which is taken by the roots can be sent out through different aspects. So one we talked about is guttation. So try to remember what is guttation, etc. So then it will be easy. So root pressure, right, provides a modest push. No, it will not provide a full push of the water right from the roots to the all the parts of the plant. Then what happens is the majority of the water is actually transported by or translocated by our transpirational pull. So this is negative hydrostatic force, which actually acts on what do you say? Are you able to follow, guys? So this transpiration pull can be explained by cohesion tension transpiration pull model. Okay. So what exactly this cohesion, addition, and what are the cohesion tension transpiration pull theory? We'll let's see about it, and we'll wind up the session as soon as possible, right? So this transpiration can occur, or it's a phenomena where the transpiration pull, or it can actually transport the water from different heights, right, from the source to sinks to source when we talk about the phloem. But here when we talk about the xylem, at the same time the water transport, we'll be talking about the uh, you know pushing or pulling force that we will talk about. So most of the researchers actually agree that the water is mainly pulled. It means negative hydrostatic force. So, the suction type of a force is hydrostatic force is acting upon the within this vascular tissue, and it is pulling so that it is able to move to the larger distances, right? So what is the driving force for this entire transpiration is that cohesion tension transpiration pull model. So what exactly this cohesion means? Cohesion means mutual attraction between two water molecules, right? So when there are two water molecules, mutual attraction between two water molecules. So that is cohesion. So cohesion actually. So this is the first one, that is cohesion, right? And the second one is adhesion. Adhesion is attraction, right? Adhesion is attraction. Attraction of water molecules to the polar surfaces. Attraction of water molecules towards the polar surfaces. So it tends to attract, right? And the next aspect is surface tension. What is surface tension? Water molecules are attract to each other in a liquid phase more than in the gas phase. So they tend to attract in more in a liquid phase than in gaseous phase. So this is surface phenomena, surface tension. So the more in contact, it would be, the movement would be that much less. So this cohesion adhesion would actually, what happens is, it negatively resists the movement of the water, but it would help in filling the gaps during this transpirational pull. So negatively affects the transpiration actually. This cohesion tension actually. So the adhesions, right? So in this, yeah. So actually, these three properties gives the water high tensile strength. That is, ability to resist the pulling force, and high capillarity. It means it is able to raise within a thin tubes. So within the thin tubes, the water can raise because it is able to have a cohesion between the two water molecules, and adhesion between water molecules, and attraction towards the water molecules, and tracheary elements. We would have xylem tissues or different types, xylem other tracheary elements, etc. So that is something which is hydrophilic in nature. So they will be attracted to this sort of uh, polar surfaces. And surface tension is that water molecules are attracted to each other in a liquid phase, right? So this gives a tensile strength, in the sense they resist the pulling effect, but at the same time it gives a capillary effect, right? It means it is able to raise within the thin tubes. In plants, the capillary it will be aided for the small distance travel of the water. Okay, so that is about this cohesion tension, cohesion tension transpiration pull theory, right? So these are the important definitions that you need to focus. Okay, great. Right. So if you see this, what else we can understand here is how the transpiration would getting affected, right? So what are the different types of transpiration? Transpiration can occur through different parts. Stomata is the main way transpiration takes place. Again, the stomatal opening and closing, again based upon the molecules, this water potential and solute potential, ionic, osmoregulation and concentrations only, the stomata pore would be closed and open. This is flaccid and this is turgor, right? So that is, you need to just focus. That's self-explanatory. And you will have lenticular cells also, where to this lenticels the excess water will be moved outside. Cuticular transpiration, stomatal transpiration at the same time through the bark, right? And advantages once focus, and the transpiration is affected positively by relative humidity, right? Negatively by humidity. If it is more humid, now transpiration would be less. Temperature, if more temperature, transpiration is high. If more light, transpiration is high. More wind, transpiration is high. Soil water is high, transpiration is high, right? We think it is directly proportional to these factors, external factors. when come to internal factors like leaf surface area. If the leaf surface area is more in monocot leaf is isobilateral, right? So you will find stomata on the both the sides. So at the same time, leaf surface area is more number of stomata, transpiration is more. And you have sunken stomata, transpiration is less. Thick cuticle, transpiration is less. Mesophyll, presence of more mesophyll cells, transpiration is less. So you need to focus on how the transpiration is positively affected, negatively affected. That also you need to touch. Okay, and then we'll enter into the now mineral transportation of minerals, right? That's self-explanatory. This phloem, when we talk about the phloem, that is pressure flow or mass flow hypothesis, where here in this phloem only, xylem is very simple. Xylem is one-way traffic. Here you need to understand phloem. Within the phloem, so if you take movement of minerals and translocation of food materials within the phloem, right? So within the phloem, what happens is, so phloem, if you consider xylem, water direction, water movement direction, it is from roots to parts of the plant. But when it comes to phloem, I mean the food particles, food substances, minerals, and these nutrients, right, and other organic compounds, how they will be moving? It is bidirectional. Why bidirectional? Because there is always a source and sink. So you'll be having a source. This source and sink can be interchangeable. How? Sometimes most of the time source would be a leaf. In leaf C6H12O6, glucose is prepared, and this glucose after polymerization or this in a more storage form, it becomes starch. Okay, cellulosic starch, right? The starch it has to go to the sink. Here sink is storage parts, right? So the food material needs to move from the source where they produce and the sink where they are storing. So this is one way movement, and the other way movement is that sometimes the sink and source also would be reversed. How, in what condition is that? Sometimes the stored food materials will be again utilized by the plants. For example, if the dead leaves are there, if they are about to dead, they will fall. The all contents would be entering into the neighboring leaves. Or if there is any damage or if there is any food resources that needs to be utilized, then the storage, or the one which is stored this food material, acts as a source, right? And the body part or the plant part which needs that particular organic compound would act as sink, right? So the sink and source can be interchangeable depend upon the situation. So that's why this bulk movement within the flow is a bidirectional, and it acts through this by mass flow, right? So mass flow is one of the aspects that you can you'll see that translocation of mineral nutrients and ions. At the same time, food particles, especially the food particles, would be taking place. So in this mass flow or pressure flow hypothesis or theory has, what it will state is that according to this hypothesis, the organic solute translocates into three following steps, actually three different steps. Okay, one is phloem loading. So phloem has to be loaded from the source, right? So when the phloem loading is an active transport mechanism, because you see that it involves energy consumption to actually move or translocate this food particles, right? So it is carried out by specific carrier protein now see, everything would make sense. The entire next half of the lesson is completely depending upon the first half of the lesson, that morning and before we talk. Now this would be becoming self-explanatory, right? You can, once you read, it will be very easy also. Okay, this is not first time you guys are reading, so and I'm trying to explain within the time frame also. Okay, so by using this phloem loading, the phloem would be loaded, it means by using the ATP. So this energy is obtained from the photosynthesis by the means of your cells. So here transportation occurs to the sieve tubes by the veins of a leaf. So the sieve tubes which are present here, right, they are closely associated with the veins of the leaf, and then they will be loading the phloem. Phloem loading is the first step, and then long distance transport where the sucrose in the stem and the root phloem. So it would be moving right from the stem to the root for the storage purpose. So what's happening here within the phloem? Pressure is creating, right? Higher pressure to lower pressure, it is trying to move. It's like Amazon. When we click the order, the Amazon products would reach from the source, and they will try to enter into the sink, my room, right? Once they reach the once the order is placed, first they will phloem load into their respective vans, and then there will be long distance transport will be there. Second step, it means they will have to move from Chennai to Gobi, and then unloading. They have to unload the products that I ordered, phloem unloading. So now what happens is pressure differences would create. So always this phloem would unload from higher pressure to the lower pressure. Once this phloem is unloaded, right? I mean this unloaded at the roots, right? It means it is again a passive transport because higher to lower, na? So once the sucrose within this phloem is unloaded into the what you say, the sink, nothing but the storage spots, now the pressure difference would be created. So when the pressure difference is created, what happens is the water tends to enter into the phloem. Water tends to escape into the phloem. Okay, so in this way what happens is this movement will be taking place. The phloem would be unloaded. Right, did you understand this concept? So once this what do you say, did you understand? So once there are three different steps involved in this phloem. First phloem loading, okay, and long distance transport, and phloem unloading. So this creates high pressure to low pressure. So this movement, bulk flow would be, mass flow would be taking place from higher region to lower region, right? So even this will be mixed with water, because this phloem would contain more number of solute particles. So water tends to move to the phloem, because phloem has more solute particles. So higher solute particles, less water potential, and xylem elements are closely associated with phloem. So xylem has more water, so more water potential, higher water potential to the lower water potential. Water tends to move towards the phloem, and it will create a passage. So likewise, the phloem would be, phloem sap would be moving to the storage areas, and then once it is unloaded, finally what happens? All the solute concentration is unloaded, right? Then what happens? Less number of solutes, more water potential, and again the water tends to enter into the xylem vessels. Like this, the movement of food particles would be taking place. I hope you understood very well. I tried maximum within 2 hours. I tried to complete this entire lesson, and please do understand that is this lesson for your junior cycle for a week. It is a week stress with week stretch at the same time with some videos also. But I'm keeping your time in mind. I took the help of board also and tried to explain to my best of my knowledge. So the breathing and exchange of gases, it's a self-explanatory lesson, and it's a very short lesson. Okay, in that what you have to focus is the tidal, and we have the relationships, no? That is tidal volume. You have expiratory reserve volume, inspiratory reserve volume. Those things you need to focus. You try to first finish this lesson, right? If possible, tomorrow I'll take up that lesson, breathing and exchange of gases. Okay, thank you for attending the live, guys, and thank you for interacting also, and see you in the next class. Tomorrow we'll meet in the same time, 9:50 to 10:50. Mostly I'll try to cover that lesson within half an hour. Okay, meanwhile you touch those areas: pressure, partial pressures, concentration of CO2 and O2 at the tissues, at the xylem vessels, etc. That you need to focus. Okay, thank you guys. Stay home, stay safe. Bye.